Yeah, welcome back everyone. We're starting the second tutorial GAN course. This course will be related to fabrication of kidney-on-chip apparatuses, and we will be talking specifically about the method of microchip fabrication method that was applied within our university. Membrane material selection and achievement of the biocompatibility and renal replacement therapy. So, since we are talking about the implantable artificial kidney, I would ask the question to the audience: what's, in your opinion, could be one of the main issues of the implantable kidney exploitation within the patient's body? Well, considering that we have to interact with blood. Pretty much the same issue has to be addressed if we're working with the artificial heart apparatuses. Anyone? Yeah, blood clotting. That's exactly one of the main issues that have to be addressed first and foremost. It's especially critical when we're talking about the artificial heart systems that have to pump blood, and they have basically the main idea behind those apparatuses is very close to what is tried to be implemented in terms of implantable artificial kidney. Heart failure and chronic heart diseases are even more critical for a patient because the primal system that provides the basic functions in the human organism is basically the lung system and the cardiovascular system. And if there is a heart failure and the heart stops beating, of course it's an issue when just the patient can die from that particular circumstance. So, if in case of chronic kidney disease, there is still some time when the urinic intoxication develops, and we have a very long window of opportunities to help patient and to perform the renal replacement therapy in case of the heart failure, and if there is the sudden heart failure and the heart stops pumping blood, it's an instant death for a patient. So this particular issue is even more crucial in case of the healthcare. And basically, if we talk about the issues about artificial hearts, if we talk about the stationary units that provide the artificial blood circulation, they also remove the social mobility for the patient. So the main idea behind this particular technology is the same. We have the lack of the donor organs to provide all people who have developed the chronic heart diseases, and the only way to help them is to artificially provide the blood circulation within the system. And the most desirable approach here, besides the organ transplantation, of course, is the development of artificial heart and total artificial heart that can maintain the blood circulation within the human body. Considering the fact that when we talk about the implantable artificial kidney, we basically have the same issue as the artificial heart, because we have to consider the blood clotting in the system, and we have to develop the measures how to prevent the blood clotting within on the internal surface of the apparatus. So, speaking of the different approaches, like the most obvious but not very desirable approach would be the heparin infusion. The long-term heparin infusion can lead to different issues with the patient's health. So, the solution that has to be applied in terms of the artificial organs like artificial heart or artificial kidney, and probably the audience can suggest what kind of solution we can propose here. If we have no option to constantly infuse the heparin in the blood system, and we have some part of the device that interacts with blood, what could be done to prevent clotting within the system?
External surface of the device could be coated with some human binding.
Yeah, that's actually that's a correct answer. So we have to consider a type of anticoagulation surface that will prevent the mechanism of the clot formation within the internal surface of the apparatus. In terms of the heart, it's of course the pump itself, and in terms of the implantable artificial kidney, it's different microfluidic systems that provide the initial blood filtration. It's the microfluidic systems that provide the reabsorption from the ultrafiltrate to the blood. So we have a lot of parts of the implantable kidney that have a direct interaction with the human blood, and of course we have to take into consideration that this issue has to be resolved by application of the anticoagulation surface to the internal surface of the device. So basically, if we talk about the types of the surface that actually main groups of the anticoagulation surfaces that can be applied is and the phylizing surface that basically uses, for instance, the vascular endothelium as the surface of as the white tissue that deposited on the internal surface of the apparatus. It could be the biomimicking surface. A type of artificial film or artificial surface that basically imitates the biological surface of our internal system, and basically that could prevent the further growth or further formation of clothes. And it's a bioinert surface. Basically, it's a type of surface that does not interact with the components of blood in any way, that allows to prevent the clot formation at the early stage of the, right at the beginning of the blood formation. So, yeah, that's just another quick overlook of this apparatus. And when we're talking about the blood formation, this is one of the potential mechanisms that actually take place during the blood formation. And if we talk about the bioinert surface, what we're talking about, we need to take into consideration what actually happens during the thrombus formation when medical device comes into the contact with blood. So the biocompatibility is actually a very huge topic. I think you can form like another course of lectures just covering the problem of biocompatibility. It's not just the immune reaction, it's not just the some inflammation processes that might take place in during the exploitation of any medical device. It could be the blood, the problem with thrombus formation as well. Because, for instance, if we take a titanium, a typical and a common material that is used for the production of the medical apparatuses, such as the pump for artificial heart, it's considered to a biocompatible material. That means that if we have the direct interaction of this material with the skin, for instance, there will be no reaction with that. So we have pretty much a lot of biocompatible materials, like the controller that I hold right now, or the watch that I'm wearing. Like there is no any allergic reaction or any inflammation that is could take place during the interaction between skin and my watch. But the biocompatibility, as I said, is a broader term because it depends on what exact condition we exploit this material. Because one thing is when I just put a watch on my hand and there is only interaction between skin and the material itself, and there can be interaction with the blood. And the blood is a very complicated tissue of the human organism, a connective tissue that basically has a very wonderful property to distinguish between the tissues that are natural to us, that are our own tissues, and any material that is alien to our organism. So even if we consider the most biocompatible materials we can think of, like titanium, or even more complicated materials that basically should not create any reaction, the blood actually recognizes that this surface of the medical device is not our own. It's not the blood vessel, it's something else. And that initiates the blood clotting mechanism. And one of the one of the mechanisms that take place to the clot formation is basically can be divided on the following stages. So the first stage, we have the adhesion of the first protein layer that consists of the small size proteins. So they're basically talking about the albumin. So this leads to the adhesion of these albumins to the surface layer. Then on the second stage, there is a displacement of the first protein layer by high molecular weight proteins. And that basically leads to the reversible changes of the proteins. So there is a modification of these big proteins like fibrinogen that then interacts with the platelets, and that leads to the activation of the blood coagulation by means of the activation of the adhesion of the platelets. So that's basically one of the mechanisms that leads to the thrombosis formation. And the wonderful property of our blood, just like I said, no matter how hard we work on biocompatibility of the device that we want to present, the organism always has to means to recognize that something is off, if something is not right. So if something is wrong, the organism will start dealing with that. So the main idea of the bio-inert surface is to provide a sort of film that prevents the thrombus formation right at the beginning stage of this particular mechanism. So we prevent the adhesion of the small protein layer that actually activates the next stages of the thrombus formation. And basically, that's the main idea that lies behind the surface preparation for the blood system circulation. And the same sort of idea, if we talk about the biocompatibility and about the safety of the apparatus, including the implantable artificial kidney, we have to think about what kind of the film we can provide in order to deal with this type of reaction. For this particular research, a microfluidic system was created. It was manufactured by our colleagues, specifically by a very prominent scientist and engineer, Kristina Popovic. She is working on the issue of the blood clotting in the artificial heart system, but considering that in terms of the interaction with blood, implantable artificial kidney and artificial heart are very close types of apparatuses that experience basically the same issue that has to be addressed. That means that basically the microfluidic system that is created for the purposes of the heart improvement, could be potentially used for the formation of the microfluidic systems that can address the problem of the blood clotting. This is like the type of microfluid that I will just allow everyone to look at that close. So don't worry about this film. The only request I have, please do not break the microfluid system itself. The film is just the experimental, so if you just break, it's just a glass substrate with the experimental film for this particular device. So the main idea, of course, we need to prepare a specific film that will provide the implementation of the idea that we will prevent the formation of the first surface layer. And in this particular case, there was a preparation of the collagen solution that included carbon nanoparticles. Specifically for that particular reason, were chosen multi-volt carbon nanotubes with different concentrations. So the problem with carbon nanotubes is that depending on what type of carbon nanotube we're talking about, we have to take into consideration that different types of carbon nanotubes with different properties can possess different biocompatibility issues. For instance, if we're talking about the single-wall carbon nanotubes, their properties can change to chirality, the way how the carbon nanotube spins and what symmetry it has. And this type of symmetry can change the electroconductivity properties, it can change the mechanical properties of the carbon nanotube. And basically the main issue with the single-wall carbon nanotube is that it has the penetration property, that it can go through the membrane of the cell and it can potentially damage the biological tissue it interacts with. So despite the fact that it has very amazing mechanical properties that are quite suitable for surfaces that we're going to use in this system, the safety issue is the primal concern here. So if we're talking about the carbon nanotubes, basically the main candidate for the further experiment is multi-wall carbon nanotube that basically does not the same interaction with biological tissues as the single-wall nanotube, carbon nanotube. And considering why exactly we use the carbon materials, nanomaterials here, is the issue that it is one thing that we actually obtain the bio-inert surface. Another thing that we actually constantly, continuously exploit this surface in the apparatus. So we need to make sure that it's resistant enough to the blood flow that goes through the device. So this particular surface will not degrade, because the more it degrades, the bigger chance that at some point we'll initiate the first stage of the blood clotting. And if there is no any surface that prevents that, then this surface can be gone due to the fact that the exploitation time is so long that blood just erases this surface. We need to take into consideration the mechanical properties of that surface. And one of potential candidates that can be used for the formation of the inert surface that can be used in the apparatus is basically the biomaterials, like combination of collagen with the multi-volt carbon nanotubes. And why I'm talking about the mechanical properties and taking into consideration that we're talking about implantable artificial kidney, considering that we talking about the shear stress of the flow of the blood flow, usually in the apparatuses artificial heart, it will vary from, if I remember correctly, from somewhere from 0 to 120 pascals. We have to take into consideration that we have the shear stress change somewhere in the same values maybe will not reach the 120 pascals in the system that we present for the implantable artificial heart system. Nevertheless, we have to consider that we will create a shear stress that can potentially degrade the material that will work inside of the microchip. That means that we have to prepare the solution that will be deposited in the substrate or the internal surface of the apparatus. So the film preparation actually had the following stages. First, we bombarded the substrate with the titanium nanoparticles. Then there was the preparation of the surface to make sure that there will be a better adhesion for the multi-wall copper nanotube collagen solution that we prepared for that purpose. There was a preparation of the solution itself. So the problem with copper nanotubes is that when they are not functionalized, they have the tendency to actually merge with each other by creating the carbon clusters. It's basically a formation of different carbon nanotubes that they let's say merge together. It's not very accurate word, but the main idea that if we do not functionalize them, they will interact with each other. So it will be a harder issue. It will be much harder to actually make a homogeneous solution that will provide us a better distribution of carbon nanotubes of the film in the film. Because for the proper mechanical properties, we need to make sure that there is an even distribution of the carbon nanotubes in the film that we use for this particular surface. So there was a functionalization of the carbon nanotubes, and the final solution that contained the 0.0 weight percent of the carbon nanotubes used for the further distribution of that film across the substrate that we've just prepared at the first stage. And this is basically how the samples look like. So we have the borosilicate glass as a substrate with the deposited titanium film and deposited layer of the bioinert surface that basically should work as the internal surface of the apparatus. So it's not just the titanium like in it's a modified version of the internal surface of the apparatus, and those samples were used specifically in the microfluidic system to test out if this particular surface is resistant enough to the shear stress that would create within the apparatus. And the microfluidic system is basically very desirable because it's very easy to use. We can easily change this substrate we have to use, so we don't have to use very complicated big systems like the typical pump systems where we cannot create a specific shear stress and control the shear stress at the whole peripheral surface of the apparatus. So microfluidic chip in comparison with conventional methods has very an easier control of the shear stress within this microfluid channel, and basically we can change the substrate or we can just extract this substrate and observe what kind of processes that took place in there. And this microchip can be potentially upgraded for the microfluidic system that can be used for implantable artificial kidney device. So basically this particular microfluidic chip was of course made in terms of the test for artificial heart, but the basis of this technology can be further developed for the microfluid chips that can be worked for organ on-chip manufacturing for implantable artificial kidney. So the main idea was to create the proper substrate and the proper microfluid system with the microchannel for the circulation of the fluid within the microfluid chip. And that's basically a plate based on polydimethyl siloxane with the form channel within. So there was basically a scaffold for that. where we just created the form for the microfluidic flow that we wanted to implement inside of this particular material. And then we just added the material that created a specific form of the microfluidic channel. And that's the experimental surface that we're working with. And in order to create the hermetic properties to make sure that there is no leakage of the liquid or there is no contamination with different gases that actually dissolved in air, we need to make sure that there is a solid connection between the microfluid channel and the surface. So basically that's the first the initial implementation of this system. So we have the fixation of screws that basically create the pressure hard enough to make sure that there is no leakage of the solution. And there are pipes to acquire the constant circulation of the fluid within the system. Yeah, this is basically the idea how the channel itself was formed. So there is a specific mode for two-component solution of molybdenum dioxide that was created by means of the 3D printing. So there is a form basically that provides the formation of the microfluid channel. So when we actually add the polydimethyl siloxane inside of that form, and when this particular material becomes solid, we just remove that form and we acquire the microfluid channel with the properties that we need for the experiment. For that purpose, to make sure that to test the fields that we want to use as the potential candidates for bioinert systems, the following test equipment was assembled. So basically we have the microfluid circulation through the microfluidic channel. With the entire system was actually hermetic, so we needed to make sure that there was no leakage or any contamination of the system within the circulation unit. And there were different equipments that controlled the direction of the flow and the type of the flow that we experienced during the whole test. So we have the opportunity to control the velocity of the flow within the microfluid system, and we controlled what kind of whether we have the unstable flow or laminar flow inside of the microfluid channel. So it helped us to evaluate what particular shear stress parameters we experienced during the exploitation of the microfluid channel. Because of course if we have somewhere unstable flow, it will create a specific shear stress, and that can affect the surface that we want to use to prevent the clotting. And yeah, for that reason we used the solution that imitated the blood in terms of the pH value and the albumin concentration. And the albumin was chosen as the type of the small size protein. Just to remind the main idea, the first thing that we have to prevent is the formation of the first protein layer. That's the small size proteins, usually albumin, that initiate the further stages of the blood clotting. Excuse me, my nose is a little bit stuffy. For that reason, we used the phosphate buffer solution that actually provided us the accurate value of the blood pH is about 7.3 in that case, and we had the concentration of albumin that varied somewhere between 20 to 40 grams per liter. Basically, it's the range that can be absorbed in a human blood. And on these stages, what was the main advantage of this particular microfluid system? It's very easy to assemble, so we can stop the experiment at any stage we want to control. So if we need to control the integrity of this surface, we can actually disassemble it every single hour and check what particular processes take place on the surface of the film that we're using as a test subject. And basically those are just different types of the films that were used in this experiment, and the different concentration and different compositions of this film basically provided different results. So the best result was achieved for collagen multi-volt copper nanotube with additional substrate that increased the resistivity of this surface to the shear stress. So in terms of that tutorial, basically the main idea is the manufacturing of this microfluid channel, and that's basically this microfluid channel provides a better an easy to use experimentation process that provides to the shear stress, and whether they provide an adequate yeah, for further reading, I strongly recommend the article provided. Basically, this tutorial was based on the experimentations that they did in the laboratory, as the stability and homogeneity. Observe these particular issues because one of the main biocompatibility issues, if we talk about the perspective of this particular tutorial, is not just like the inflammation process issue here, and that's a part of biocompatibility as well, and we have to take this into consideration. So in terms of implantable organs, including implantable artificial kidney, is to prevent blood formation, because it could be a crucial issue in terms of the implantable artificial kidney exploitation. And of course this with editors like Professor Kolchik Gucha and my intention, I'm sorry. So yeah, for better outlook for this particular tutorial, you can address this. If you have any question in regards to that tutorial, I'll be glad to answer them.
Thank you for the lecture.
Yeah.
Three kinds of anticoagulant surfaces.
Mm-hmm.
Do they have response? Do they elicit similar immune response from the body? I don't have much background in biomedical sciences, but as a layman, I would say have less, or let's say milder immune response.
Immune response, if we are talking about inflammation, etc. All these three types have in terms that they do not provoke any immune reaction, but in this particular tutorial, anticoagulation surface material. So we were not talking about the immune response, but the mechanism that was considered in that case. And biomimicking means that the endothelial surface is basically a biotechnology where we make that basically is the like a major, so the blood will not proceed with the blood clotting process on that particular. Considering it can be a desirable option, it's still a very complicated. We have to consider the immune response in that particular case, because even if we, by means of biopsy, there might be a chance of the immune response, but basically why the types of cells in our organism, and blood actually recognize that it will not actually mimicking surface, from my understanding, is an attempt to that we have inside of our organism. So we are trying to mimic any position and the way how the blood will react. Biotechnology, of course, is not my direct field of expertise, but there can be some probability that there will be a clone formation even on biomimicking surfaces. And do not look that exciting as the biotechnology, for instance. The main idea version of the protein layer to prevent the other stages of the basically the main issue, the main try to resolve with this particular anticoagulation surface is to prevent the blood clotting. Strategies for this, and the core difference between those two steps and those two inert surfaces it has to prevent the formation of the initial other stages of the blood clotting. But of the immune response, it's just a different topic, and it requires a different type of exercise. Any biological tissue will not provoke any type of immune response. This is just a small part that we covered, that a small issue of biochemistry here. Yeah. Thank you.
What is the cost associated with the
cost of manufacturing?
Yes, sir.
Oh, since it's a test microfluidic chip, I will not be able to properly estimate the full cost of the process. The main issue here is to acquire the domestic products that can be used in the manufacturing, because if you order some parts of the microfluidic channel from abroad, it's usually a very expensive thing. And this, considering this is a test chip, I'm not ready to provide an economic evaluation of how much budget was spent on manufacturing this particular test chip. But the main concern is the acquiring of the domestic products, because it's usually a big issue if there is some economic sanction, or the manufacturing of specific object can be blocked by the sanction that prevents some components to come in.
Thank you, sir.
More questions? Yeah, so basically tomorrow I'm going to summarize just the main ideas of the selection course. There will be another very diluted lecture about the AI application, and then we will observe, or rather review, the current prototypes of artificial kidneys that exist in terms of implantable artificial kidney technology. So we cover some biotechnological approach, and there is one prototype that does not exploit the biotechnologies. I will try to cover that during the final lecture as well. And I will try to provide the literature that I used during the formation of that course, so you will be able to address it outside the course if you're interested in some particular topics that you will want to explore further. Yeah, and that's basically the end of my tutorial. If you still have any questions, I will be glad to answer them. Yeah.
Sir, you have mentioned that the microfluidic chips are mostly based on collagen and carbon nanotubes.
Yeah.
So, what basically the advantage provided by the collagen? Because I understand it is basically a structural protein.
Yeah. So from what our colleagues were trying to do is to achieve the basic biocompatibility property of this particular film, and multi-walled carbon nanotubes are basically the type of carbon structure that provides the mechanical resistivity of this particular film. So we were trying to implement the technologies that basically would be biocompatible with blood, and at the same time it had enough of resistivity to the shear stress of the blood, so it would not disappear. It would not be erased during the exploitation of this microchip.
That's why collagen is the ideal protein for this.
Well, yeah, at least our colleagues consider that a good candidate for that particular purpose.
Thank you, sir.
Yeah.
Participant have dot, you can ask. So it is end of the morning session, and second session will start at 3 o'clock. So requested to all of you come at 2:45 to join the session. And 10th lecture will be taken with Dr. S.K. Tripathi based on the direction of the kidney disease using the photonic sensor. Okay.